Hello, so today we'll be talking about the essential components of model organisms. This project was done by Victoria Fernandez, Gabriela Hinojosa, Summer Hughes, Jacqueline Ramirez, and Gabriel Cepeda, and myself, Arisla Diarriera. To start off, I will be telling you guys what a model organism is. A model organisms are species that have been widely studied for experimental advantage because of its ability to be maintained and bred in the lab settings. It also allows to understand the biological phenomena. Model organism discoveries can give insights towards the others working organisms. An example of a model organism would be a white lab rat. Because of these organisms have very similar gene sequences to human organisms, those model organisms help scientists experiment different things prior to human trials. Once it has proven to better benefit our health, the organisms then mark a medical advantage for our species.
Okay, so I'll be explaining why these organisms are common to use in lab. For example, E. coli, which is the one we understand the most. It reproduces fast, and also it's able to develop mutations in a rapid rate, and it can also be grown in a simple nutrition broth in a lab, as well as yeast, which is very similar to E. coli. It reproduces fast and also develops mutations rapidly, and also can be grown in a nutrition broth, but it also has a small genome. Zebrafish are small. They can be manipulated easily. They produce a lot of offspring, and also they have a big similarity to a human genome. Also, it has a transparent embryos, which allow us to easily see stages of the development. And a fruit fly, which is a multicellular organism and is more helpful than yeast. There's also mutations available for any gene. And the last one, which is mice, since it's a mammal, is very similar to human, and it also can express the same phenotypes as humans. And also they're all very inexpensive except for mice.
I'm going to talk about how model organisms are easy to breed. They're easy to breed because they could breed in large numbers. They have a very short generation time, therefore many generations can be reproduced at once. These model organisms are easy because they allow scientists to study certain characteristics and diseases in lab. An example of this is E. coli. So E. coli cells can double every 20 minutes, and at that rate, you would produce 1 million E. coli cells in 7 hours. And that the fast growth of E. coli means that scientists can experiment with them more frequently than other types of organisms.
Expanding on what Summer just said about E. coli, in our Bio2 lab, we were able to measure enzyme activity on a plasmid, and enzyme activity is important because it is vital for the study of enzyme kinetics and enzyme inhibition. And in lab, in 80 minutes, we were able to take a plasmid and see where the enzyme, the restriction enzymes, were able to cut the plasmid. And this is important because then you can see where, for example, the GFP protein would insert itself to give E. coli antibiotic resistance. And that's why having a model organism like E. coli is important. Model organisms occupy a pivotal position on the evolutionary tree despite their genetic or experimental advantages or disadvantages. So for example, the pufferfish has a similar gene repertoire to humans, but a much smaller genome. Therefore, it's much easier to study a pufferfish versus a human, and they are cheaper, and we also can't really test on humans. And the flatworm bilaterians occupy a pivotal position in metazoan life as they represent two separate phyla. So one study could be a study for two. As I was explaining in the last slide with jellyfish, the jellyfish genome is much smaller than the human genome, so it's easier to study the jellyfish. Right here you can see that the human genome is 7 times larger than the E. coli genome, and it would be a lot easier if we just studied the E. coli because 4,300 genes versus 30,000 genes, it's much easier to sort out the E. coli DNA.
With Gabriel's explanation about monoorganisms occupying the pivotal position in the evolutionary tree, it goes to show just how similar we are. For example, E. coli may be in the bacteria spectrum of the evolutionary tree, and we may be in the eukaryota, but we still derive from a common ancestor. And because of that common ancestor, we can use these monoorganisms to combat diseases and learn to treat or diagnose them. Scientists continue to work towards understanding all the molecular processes that underpin the human biology and health. They are currently using research organisms or model organisms to see how these animals or bacteria can further fix damaged pieces of DNA or regenerate broken bones or missing ones. The studies of these organisms can help us reveal molecular changes that are associated with diseases and show the connections between certain diseases and how they're unrelated or related.
So the reason why we use the model organisms and mutants as discussed earlier is to study diseases. So typically it takes the natural course of a disease in a human takes an extremely long time. But on the other hand, if we were to use model organisms, this allows us to study diseases in a shorter period of time. So we can have the symptoms develop in a very smaller time frame compared to humans. And so some examples of model organisms that we use are yeast, and this allows us to understand how cells divide and how cells are able to be duplicated. As for fruit flies and worms, we use this to understand the development of embryos. And as for zebrafish, we use this we can use this to understand how vertebrates develop, and the reason why is because the embryos are transparent, as you can see in this image. Image B. So because it's transparent, it allows scientists and research to see the cells moving around. And back to this slide. As we stated earlier, we used E. coli, and that can allow us to get an understanding of genes and get an understanding of how they work, and we can also use that for medicine to understand which type of medicines we can use for antibiotics, antibiotic resistance. And of course, we use mice to be used to test for drugs, and so to be used to test for.
So to finally recap on what we have discussed previously, a model organism. What a model organism is, they are species that have been widely studied for experimental advantage because of its ability to be maintained and bred in a lab setting. Common organisms that are used are E. coli, zebrafish, a nematode worm, yeast, fruit fly, and mice. To continue on, they are easy to breed. They're easy to breed because they breed in large numbers and they create many generations at one time. For example, like we've given before, E. coli, which doubles every 20 minutes. Another thing is how they relate to the phylogenetic tree in relations to humans. Because of their ancestral connection, way back when, we all have a common ancestor, which connects us in a biological way. Now, model organisms are used in everyday life by researchers and scientists to study disease and how these diseases can be treated and diagnosed in humans. These are model organisms' purposes and uses for essential life.